On. There we go. Being an entomologist, I have been asked a lot about this insect or that insect. Most of my presentations, for those of you who have seen them or maybe seen them on YouTube, I talk in generalities and I talk about general things of insects. But every once in a while, I get asked to do a deep dive. And this deep dive sometimes deals with a particular insect, where I spend an entire hour talking about one insect. This one is a little bit different. A little bit of excitement too, because sometimes I get questions about centipedes, millipedes, spiders, scorpions, ticks, and things of that sort. And so now I'm looking at a mite. Technically, I'm not qualified to talk about this because I'm an entomologist, someone who studies insects. However, I was asked to do this a number of years ago. This is something that I completed 3 years ago. And when I was asked to do this, I thought to myself, I think I can do this. Because it was in 2016 when I deciphered the insect olfactory code. I was having some degree of success with the insects, and then I was asked, can you do a mite? So inside, I was thinking, I don't know, I don't know. But outside, I said, absolutely I can. Sure, it's not going to be a problem at all. And so when I got thrown into this, I didn't really know what I was getting into. But now that it has been completed, it has been an absolutely wonderful journey. This is going to be a little bit different than some of the talks that you have heard me give before. Some of you are going to absolutely love this talk. Others, not so much, because I'm going to be throwing more science into it. I am going to be talking a little bit about frequencies now, and so there's may be a comfort zone for some. This may be a bit much for others, but I'm now going to move in to give you a true deep dive of the two-spotted spider mite. What's my goal? My goal is to make sure that you are more knowledgeable than you've ever been about the two-spotted spider mite by the time I finish this presentation. So, I call it the two-spotted spider mite, likes and dislikes. It's going to be a chemoreceptive analysis, where I have the ability to take a look and see what the insect is smelling or tasting, what it likes, and what it dislikes. And that will be clues for you who are battling with the two-spotted spider mite. Okay? So first I need to begin, what is it? It's not an insect. It's class Acari. And so, yeah, we do have the Mesostigmata over there, 10,000 species, a lot of predatory mites. We've got the ticks, which are in a different group. But off to the right, I've got the superorder Arariformes, the Prostigmata, which are the sucking mites, not the predatory ones. And then we have the family Tetranychidae, which are the spider mites, about 1,300 species. I'm not talking about 1,300 species. I'm only talking about one, and that's going to be Tetranychus urticae. Tetranychus urticae is the two-spotted spider mite. It is the most prevalent mite in agriculture today. It is by far the most destructive one, and that's why I'm going to be spending this time talking about one species to make sure that I'm going to be able to answer, hopefully, a number of the questions that you have, since this mite has been a bit of a mystery.
Sir?
Yes?
Yeah. What's the difference between an insect and a mite?
The difference between an insect and a mite. Insects have 6 legs. They have a number of other features that are associated with them, 1 pair of antennae. The mites do not have antennae, and they have 8 pairs of legs. They also have 1 body part. And so generally, this one body part, although the head and the abdomen, the cephalothorax can be associated with one another, but there are some differences between them. I will allude a little bit to some of the differences, but just so you know, that's because they have 8 legs, some people consider the mites and the ticks to be closer to a spider than an actual insect. But they are different. Another big difference, most of the insects have wings. The mites do not. And because they don't fly, this changes their dynamics a lot. So here's a picture of the two-spotted spider mite. You can all see they have two spots, thus the name. So if you're looking under the microscope, because you're not going to be able to see the two spots, you could barely see the mite itself, but the two spots, you really got to be under the microscope because they are very, very tiny. All right, I've got a number of talks on insect olfaction. For those of you who have been through those talks before, this is going to be a little bit more understandable for you. If not, I'm going to have to tell you what I'm doing. But if you wanted to, you could go through and check out on the internet problems with the current theory of insect olfaction, or toward a new theory of insect olfaction, or toward the deciphering of the insect olfactory code. And then I do actually talk about aphids as well. So this presentation is going to be somewhat similar to the aphids, and it is going to talk about some of the same spectrums that you may have seen before. If you have not seen any of these presentations before and you suffer from insomnia, these talks will cure you. All right. Insects only feed upon food that is considered unfit, nutritionally poor, dead, or dying. If you've never seen this before, you need to see it now, because you need to look at insects as not as the enemy, but as an indicator species, which is what I alluded to when I was sitting down with Glenn recently. Here's the chart. This is the chart which I've talked about in many presentations. You will notice, and I need to point this out, check out the top, leaf Brix, not fruit Brix, not stem Brix, not root Brix, leaf Brix. We are testing the leaf. When you are testing the leaf, you can use this chart because they almost never break 20. Once the leaves get that high, they start dumping sugar to different parts of the plant. So I stop it at 20. But if you are looking at an unhealthy plant at 1 to 2 Brix, that's the range you're going to be in. From 3 to 7, things start to pick up a little bit as far as Brix scales. But take a look at this. What happens at 6? Plant secondary metabolites increase. Pay attention to that right now because that's going to be coming up later on in the presentation. Once you hit 6 Brix, the plant's secondary metabolites start to increase. Those are the things that add color and taste and smell to the plants. So if you've ever had a vegetable before that tastes like cardboard, that's an indication that it's going to be below 6 Brix. From 8 to 12, resistance begins in earnest on the plants. Water retention abilities increase drastically, and by the time we're getting to 12 to 20, especially 14, 15, 16 Brix, you don't have insects and you don't have disease anymore. Now, this is an interesting chart in the sense that I've actually taken the feeding guilds of various insects. I've got the aphid group. The aphids include aphids, they include scale insects, they include mealybugs. This aphid group is not interested in eating a plant when it gets to about 6 Brix, 7 Brix, or 8 Brix. The next group, the sucking insects, of which the two-spotted spider mite should be one of them, is at about 7, 8, or 9 Brix when they start to lose interest in the plant. The chewing insects, like caterpillars, 9 to 11 Brix, they will actively lose interest in a plant. And then finally the grasshopper group, which includes the grasshoppers, the katydids, the crickets, things of that sort. They're going to lose interest by the time you get up to 10, 11, or 12. That's the insects. What about the mites? I didn't know. When I began the investigation, I didn't know where the mites were. Has anyone ever looked at it before? Because I actually had not. I'd spent time looking at the insects, but not so much the mites. Found a 2023 paper which found that the two-spotted spider mite prefers, not that it can't, but it prefers Brix readings between 1.6 Brix and 2.6 Brix. So for those of you who want an answer as to where the mites are, it's really low. Apparently, even further below than the aphids. And this is a bit surprising because when you have a 1 Brix plant, it looks terrible. And a 2 Brix plant also looks pathetically bad. So how is it that the mites are interested optimally? They'll certainly go after a 3, 4, 5 Brix plant, no problem. But optimally, they're looking for something between 1.6 and 2.6. How does that work? How is that even possible? Because most of the plants that they attack look much better than a 1 Brix plant. Well, I will be happy to tell you. So we're going to do this analysis. I'm going to be talking about chemoreceptors. Chemoreceptors are the proteins that allow them to taste and smell. Smelling, olfaction, refers to the olfactory system, the olfactory receptors, also known as the ORs. Insects have them a lot. We investigate them a lot in olfaction. The mites, 0. Most of the ORs are associated with flying insects. Mites don't fly. Mites don't have them. They're not flying around looking to zero in on a plant. That was the first shocking thing that I had to deal with when I began the analysis. The next one are the GRs. These are the gustatory receptors. Taste. Anytime they're tasting something, it's a GR that is tasting it. Well, usually, they're in the 50 to 70 range. I have 430 GRs. Holy smokes. This was completely off the chart with anything that I've dealt with before when I'm dealing with insects. 430? I begin to wonder if they can taste everything that is on the planet. And indeed, they have over 1,000 hosts. So they can taste just about anything. I'm going to give you some clues as to what they are tasting. The IRs, this is not something I'm going to be discussing much today. There's only 19 of them. These are ionotropic receptors. And then we also have a brand new receptor that the insects do not have called ENaCs. And the ENaCs are epithelial sodium channels. These epithelial sodium channels, I'm just calling ENaCs, and there were 112 of them for a total of 561 proteins. I have to go through and analyze every single one of these. It's a tedious process, but I had to get started. It took me about a year to get through it. These are the spectrums of an individual protein receptor for the 2-spotted spider mite. And take a look at them. There's some big peaks, there's some smaller peaks, and none of them are identical to another. They're all different. All 430 GRs have different peaks associated with them. I do have software that enables me to get these spectrums. I'm not going to discuss that right now, how that's done. But these spectrums, each one of them, I have to go through 430 times. After I do that, I have a way of combining all of them into one spectrum, such as the blue spectrum that we have right there. When I look at this blue spectrum, this now is where you guys are coming in this presentation, gives me clues as to what the mite is tasting. So, what exactly are they looking at? Well, I told you that all of these spectrums are different. How is it that they could be different? They all have different frequencies. Some of them are big, some of them are small, some of them are medium. If you are going to combine all of these frequencies into a single spectrum in order to come to some conclusions, what are you simulating? Simulating a barcode, a UPC barcode. This is how they smell. This is how they taste. You simply scan the barcode in the grocery store, and it immediately recognizes it as macaroni and cheese or whatever it is you're ordering, instantaneous. What is it detecting? It's detecting the thin lines, the thick lines, and the medium-sized lines. In addition, the gap in between each line, which is the white part, is also being measured. That UPC code contains a lot of information, and it can detect exactly what you are running across it instantaneously. That's why you quickly do this in the grocery store. Did you know that insects and mites are smelling in about 1 millisecond? 1 millisecond. 1/1000th of a second. This is how they are smelling. This is how they are tasting. It is an instantaneous effect. And when all of these frequencies come together, the mite will immediately identify what it's supposed to smell or what it's supposed to taste. This is the barcode. 430 all combined into a single spectrum. When you're looking at something like this, this looks a lot like the spectrums that I just showed you. But this one's different because this one is combined. If it's combined, that means that the more of the receptors that are contributing to a larger peak are going to be more important than the 430 that are contributing to a smaller peak. Take-home message, the big peaks are the most important. That's the take-home message. That's it. It takes me a long time to get to this point right here. But once I get to this point, I've got a lot of answers that are coming at me. So let's take a look at the biggest peak. What is that? It's 22,100. 22,100 nanometers. That's the main frequency that it's focusing more than anything else. The two-spotted spider mite is a ubiquitous mite. It is found everywhere. Everywhere. I can find it in any field of any crop. Okay? It can be found in a off-growing season, on an in-growing season. It's all over. It can be found in multiple climatic zones. It can be found infesting over 1,100 plant species. But what really floats their boat is to have hot, dry conditions. When they have hot, dry conditions, that's when their numbers increase. They're there, but they're not mating. They're not reproducing. Then, once they do that, under the hot, dry conditions, everything changes. When they are simultaneously met, not one at a time, not just hot and not just dry, but when both conditions are met, that's when things really erupt. That's when the fireworks take off for the two-spotted spider mite. And so we can go from having a few dozen to over 1 million mites on a single plant. For those of you who have dealt with the two-spotted spider mite before, you know exactly what I am talking about. But we've got this problem here. Why is it mostly preferring 1.6 to 2.6? I just got finished telling you it likes hot, dry conditions. Hot, dry conditions mean you have an absence of water. If you have an absence of water, that means you just removed one of the parts of photosynthesis. CO2 and water come together to form sugar. You take away water, you stop photosynthesis. And if you stop photosynthesis, the Brix can potentially go down very quickly. And that's when the mite moves in. So the plant may be doing pretty well, but then it's going to shoot down as photosynthesis is compromised, and the mite will then go crazy. But it's not doing what the spider does. I just mentioned, the spider is somewhat related. They all have 8 legs, but the spider is a slow eater. Once it grabs a fly, like this Drosophila melanogaster, it's going to grab it and it's going to secrete digestive enzymes into it. It's going to liquefy the body contents, and then it's going to suck it out. And this is going to take minutes, and sometimes it could take over an hour if the insect is large enough. That's not what the mite does. The mite does not secrete digestive enzymes. It just goes in there and sucks. So the mite literally sucks, both actually and figuratively. And it does not discriminate. It grabs everything, absolutely everything, unlike the spider, which is very selective. So we have an aphid digestive system here. This aphid digestive system rolls back upon itself because when certain nutrition comes in one end of the aphid, it's removed and sent to the back and is popped out the back end in less than 1 minute. So when the aphid sticks its mouth parts into the plant and it starts feeding from the phloem tissue, what's coming out the back end? Water and sugar. That's called honeydew. So if you ever parked your car underneath a tree, and you go into a dental appointment and you come back and your truck is covered with stickiness, that's because of the honeydew. There are a number of insects that are peeing and pooping on your car. It is mostly sugar water that is coming out, and that's how the aphids work. The mites don't do that. Mites can't filter it out. They have to eat it all. So here we got the mite digestive system, and that little spot right in the middle right there, that's where the food goes. It can expand, sure, but it can't get a lot because it can't filter things out. So if you have, if I asked Greg Moline, I said, would you like a double cheeseburger? And he says, yes, I would. And I said, all right. So I come out and I got this very large water balloon, and I hand it to Greg, and I say, here you go. So he looks at it, taken aback, and he sees floating inside the water balloon is the double cheeseburger that he requested. Is he going to eat it? Well, probably not. He's going to think, man, this is really gross. I can't believe you did that to me. You're welcome. And I would say, oh, it's much worse than that. I said, this is a double cheeseburger that you order, but you got to have the whole thing. You can't really pull it out. You have to take in all of the contents of the water balloon. He's like, you got to be kidding. I can't eat that much. That's too much water. I just want the double cheeseburger. And I would say, no, it is a package deal. In order to get that double cheeseburger, you have to drink all of the contents, everything that is in there. Yeah, I realize it's going to fill them up. It's going to make them feel real uncomfortable. You're starting to realize now some of the problems that the mites are running into. Mites do not like what is on the left. This is a cell that is well hydrated. But a cell that's dehydrated in a plant can go down to 90 to 95% of what it was before, which means the water's gone. That is what the two-spotted spider mite wants. It wants dried out cells, and it will suck out the entire contents of that cell right there. And it will do so in a matter of seconds. It will not do it slowly like the spider. It's just going to suck them up, and it's going to move right on to the next cell. That's how they eat. So water is very, very important for the two-spotted spider mite. So the relative volume, this is when it's filled. You have good turgor pressure. And then you get down to maybe 5 to 10% of what was before, and it's now interested in eating that. The situation has now changed for the two-spotted spider mite. So what is that 22,100 peak? Oh, it's not just that peak. It's all of those peaks. All of those peaks from 11,000 all the way up to 75,000 key in on water. The two-spotted spider mite has a high sensitivity to water. It's very interested in water. Water is a problem. As a matter of fact, it's such a serious problem that you can actually control two-spotted spider mites by having a rainstorm or heavy irrigation. That will actually deter the two-spotted spider mite because they need things dried out. and they key in by detecting water, among other things. Yes, I understand there are some peaks over here. I'll get to those in a second. But right now, we have a large number of peaks that are devoted to water. How much? 29%. 29% of the gustatory receptors are responding just to water. This goes to show you how important water is to the two-spotted spider mite and how it differs significantly with many other insects. If I take a look at the IRs, I mentioned the IRs, there's only 19 of those. We'll take a look here. We got a number of peaks over here, and they're the biggest ones too. And they're very, very big. How much of this is devoted to water? 44%. The IRs, a different chemoreceptor, have 44% of their peaks tuned in to water. I'm trying to hit you hard so you realize how important water is to the two-spotted spider mite by taking a look at its chemoreceptors and seeing what it is focusing upon. So what's the next one? Those are the peaks off to the right. Now we got the 4298. Where does the 4298 fall? That's the second biggest peak. It's almost as big as the 22,000 one. What's the deal with that one? There we go. The 4298 actually corresponds to the major carbon dioxide peak. This is a carbon dioxide spectrum. Each one of these blue peaks is a potential attractant to a particular insect. In this particular case, the biggest one right there corresponds to the 4298 peak. So they seem to be attuned to carbon dioxide. Water, okay, but carbon dioxide, why do they care about that? And there it is right there. It's actually at 4270. It's very close to the 4298, and it varies from about 4200 to 4500. Well, let me tell you why carbon dioxide is important. Right now in our atmosphere, we have about 425 parts per million CO2. How many of you know that this is not enough CO2 to raise a healthy plant? Yeah, I know there are a lot of people who know that. As a matter of fact, it's not even close. You cannot raise a healthy plant with 425 parts per million CO2. Oh, I know what you've been told. I read the same stuff you do. But in order to get a healthy plant, you really got to be at least 1,000 to 1,100 in order to get the plant doing what it's supposed to be doing, which is growing and growing well. We know back in the olden days, in the Ordovician and the Cambrian explosion, estimates run is that we had about 4,000 to 9,000 parts per million in the atmosphere. It was a different time. Plants were much bigger. They had bigger leaves. They were growing faster. Lots of CO2 to pull from. They needed to be big, and they needed to grow fast. Why? Because Brachiosaurus was feeding upon them. Triceratops was feeding upon them. Stegosaurus was feeding upon them. These are big dinosaurs, and we needed a lot of plant material, and we needed it going fast in order to feed them. That's why this was working out. We really can't support dinosaurs nowadays. We don't have the capabilities of doing that, but they did back then. All right, so what's happening down in the microbes right there? Well, in the microbes, they're breathing too. They breathe in oxygen, and they exhale CO2. And so you can have levels of CO2 in the soil between 500 to 5,000 parts per million CO2. Much, much higher than you will find in the atmosphere. That is life. That's life. But it's in the soil. CO2 gets in the stomata of the leaf. Well, that's a little bit of a problem. How do you get it up there? There are two different ways. We have micro pulsations and macro pulsations. A micro pulsation is caused by the moon. The moon has got our tides 2 times a day. It's bringing the tides in and bringing them out. This is going to move the water in the soil, and you're going to have micro pulsations which are going to start bringing up some of that CO2. A macro pulsation is a storm. When you have a low front and a high front. So if the high front comes in, it'll push atmosphere down into the soil, and they will breathe. And then a low front comes in right after that, and the soil then exhales. And that CO2 will then start to move up due to the exhalation. Once it does that, the stomata pop open. And the plant pulls as much of that CO2 as possible into its body. This makes up for the lack of CO2 that it's not getting from the atmosphere if it's a high Brix plant. If it's a low Brix plant, you're not going to have that many microbes. And if you don't have that many microbes, you're not going to get a lot of CO2 production. And if you're not getting a lot of CO2 production, you are not going to be able to feed the plant. The plant feeds the microbes through the root exudates. The microbes feed the plant by exhaling CO2 and making sure it pops up, especially when we have a low front and a high front combination. Many of us have wondered, why is it that the rain does incredible things to our crop? Part of it is because of the water. Part of it is because there is a nitrogen source in that rainwater. All that's true. But it's also because of these high and low fronts. You're pulling up a lot of CO2 in addition to that rainwater. That's why your plants look amazing after rain. They look absolutely amazing. And we're all impressed. You can't get this way with irrigation. But with a rainstorm, you can, because there are certain natural tendencies in the rain that are causing the plant to look fantastic. There's a burst of growth, it greens up, and things just look better and prettier. It's because it is. The Brix levels are going to go up. This is important to a two-spotted spider mite. If there's high CO2, it's not interested. It needs to be able to detect that high CO2 because that's going to be conducive to a healthy plant, which it doesn't want. It wants to make sure that the CO2 levels are low, and in order to do that, it has to be able to detect them. So it does. So when the stomata open up, the CO2 goes in, the Brix goes up, the CO2 is detected, as I just mentioned, and boom, they're interested. That's the 4298 peak. In addition to that, there are also others. These are CO2 peaks as well. I want to make it clear is that when I discuss a molecule such as CO2, I'm not just talking about one frequency. Remember the barcode that I showed you, the UPC code. It's an instantaneous detection of CO2. And it's detecting multiple frequencies at the same time. And that tells the mite, CO2 is high, I need to move on. And so here, in the two-spotted spider mite, I've got two main peaks, 4298 and 4484, which are tuned to CO2. I have three minor peaks, 9520, 10,473, and 15,300, which are minor peaks, all detecting CO2. This thing is attuned to a lot of things. Water's only one of them. CO2 is another. All right. 10,473. There's another big peak. Which one does that correspond to? Well, let's take a look. We've got a lot of nitrogenous compounds. And I looked at nitrogenous compounds first because when I see the two-spotted spider mite populations increasing exponentially, you can't do that without nitrogen, a massive amount of it. So we do have a lot of nitrogenous compounds. Nitrates, nitrates, nitriles, ammonia, ammonium ions, amino acids, polypeptides, proteins, amides, amides. They're all nitrogenous compounds. That's not all of them. There's a lot more too, but there's a lot to take a look at. So which one do you think the two-spotted spider mite is interested in? Let's take a look at the spectrum on the top. down below I have the ammonia infrared spectrum. There are 3 regions of the ammonia infrared spectrum. One of them is small, way over here in the corner. It's got a nice needle nose peak. This one gets a little bit bigger, and finally we get the big mama, where there's a lot of absorption in ammonia. In addition to having not just one needle nose peak, I've got 2 needle nose peaks over here for ammonia. What does that correspond to? I'd like to know. That corresponds to 10,471 nanometers. Do you see the connection? I've now got a mite which is interested in ammonia, and it's highly attuned to it. And ammonia, sometimes you can smell it when you pass by your plants. This is also what they are very interested in getting. This is going to be their main nitrogenous source because of the peak and the size of the peak. It's a big peak, and I know that they're going to be interested in ammonia. In addition, the 9520 and the 11,651 also are within this range of absorption. So we've got not just one major peak, but there are 2 side peaks, which are also enabling the 2-spotted spider mite to detect ammonia in the plant. So let's take a look at Florida strawberries, if I could. If I could take you down to a warm climate, just work with me here. Florida strawberries, if we test them, we can get some plant sap analysis from them, and we have. So we got some strawberries in Florida. What if I want to take a look at the ammonium concentration? Well, if it's lightly infested with the 2-spotted spider mite, we achieve levels of 69.9. Is that high? You better believe it is. I really don't want it to get above 30. Something below 20 would be better. 69.9 is not good. I don't like that at all, but there it is. And it's lightly infested with the 2-spotted spider mite. We went after some other Florida strawberries that were heavily infested with the 2-spotted spider mite, and their readings were 87.4. So we've got additional circumstantial evidence that the mite is very interested in ammonia concentrations. And this is testable through a plant sap analysis, and this gives you a clue as to what they're going after. Let's take a look at the next one. What is our next one? The 5700 peak. 5700 peak, which one does that correspond to? Lactones. What are lactones? Lactones are secondary plant metabolites. Remember, I asked you to pay attention at the beginning, and I told you that the secondary plant metabolites start to increase once you reach 6 Brix, and especially 7 and 8 and 9 and so on and so forth. These lactones, there's a lot of them. There's about 6,000 lactones, and they are repellents for the 2-spotted spider mite. They don't like secondary plant metabolites. They are tuned to lactones, which are among the more common secondary plant metabolites because it does not want to eat a plant that is high in secondary plant metabolites, that is above 6. And so it's keying in on the lactones. Let's see, that's pretty much what I just said. Well, let's go back to the 5700 peak. Just because I've got a line, just because I've got a peak, does that mean that's the only thing that it's detecting, just the lactones? Far from it. This also corresponds to aldehydes. Aldehydes, what's the deal with those? Those are more secondary plant metabolites. And so when you have secondary plant metabolites, lactones, aldehydes, the mite is actively detecting them. And it's not interested, and it will move on. This is all discernible just from analyzing the chemoreceptors. They are common. Hexanol is a common one. Jasminates, another common one too, but they're all vital in plant defense, stress response, communication. If you don't have these, the plant's not doing well. and the mite will move in for the kill. In addition to the 5700, there is also another peak which corresponds to the aldehydes because it's never just one. I've been doing this for 10 years right now, and there's usually about 3 to 5 peaks that are associated with every molecule that an insect or a mite is trying to detect. Okay, so that's from a 2023 paper. Part of the reason why I put that up there too, it's a Canadian group. You're welcome. What's next? Oh, yes, the 8002 peak. I never seen this one before. I didn't have it in any of the insects that I've looked at. And I went back and checked, and a few of them did have the 8000 peak, but it was very tiny. Whereas this one, big. Why is the two-spotted spider mite attracted in this? Well, I have no idea. This is the first time I've had to analyze something like this. So I hit the literature. What does 8000 correspond to? Well, one of the hits was nitrous oxide, which is laughing gas. Well, this can't possibly be correct because this is something in a dental office. Why would they be keying in on something stupid like this? So I decided to do a search, and the Google comes back and tells you that laughing gas is a dominant molecule emanating from plants. Really? Nitrous oxide. How does this work? Well, I'll tell you how it works. This is vomit. Laughing gas is vomit. Anytime a plant gets too much nitrogen, it converts it to N2O, nitrous oxide, and expels it. It vomits it out. When it's trying to get rid of the nitrogen, it does through nitrous oxide. So usually nitrates are pulled in through the roots. Usually they then turn into amino acids. Usually the amino acids can go in one of two different directions. They can form primary plant metabolites. They can also form secondary plant metabolites. This is the natural system. On the other hand, if we have too much of the NO3 in excess, and that's important, the pathway changes. It's a multi-step process that leads to nitrous oxide. This nitrous oxide is then released from the plants, and it is detectable by the two-spotted spider mite. I haven't run across insects that can smell it yet, but the mites are really good at it. And why is the nitrous oxide found in large amounts? It is only found among crop plants where a lot of nitrogen is being put down. There's very little nitrous oxide coming from plants out in the field or elsewhere, but in crop plants that are getting dumped on, there is a lot of nitrous oxide. That's what the two-spotted spider mite is keying in on. And it's very good at detecting nitrous oxide. How do I say that? Just because of a stupid 8000 peak? Well, no, there's actually more to it. Here's the nitrous oxide spectrum. And there's the 8000 peak. There are a lot of other peaks there. Well, this is getting interesting. I got a lot more peaks to work from. Are they really that sensitive to nitrous oxide, which is being caused by farmers putting down too much nitrogen? Is this actually possible? What about this peak right there, 17,000? Is there a 17,000 peak that I can look at? Indeed, there is. There is a 17,000 peak. It's at 17.9. It's a little on the high side, but there is a corresponding peak, and I just lost my screen. So I'm just going to keep talking and pretend that I actually know what I'm doing. Then we take a look at the ENACs. This is the other chemoreceptor I told you about. Look at it. What are the major peaks? It's another 17,000 peak. So it's not just one chemoreceptor. They've got another group that do seem to be able to detect it. So that's the 17,000. I got the 8,000. There's the 4484 peak. There it is, 4484, that's also detecting another frequency of nitrous oxide. What about this? We got a 3937 peak there too. And lo and behold, there's a 3969 peak. That's got the capabilities of detecting that. It gets better. So there's the 3969 peak. And then we got a 2941 peak. And if I go to a different part of the spectrum, you haven't seen this one yet, we got a 2944 peak. It's not a very big one, but it's still detecting nitrous oxide. And now we have the 2857 peak right next to that. And lo and behold, we have a 2862 peak. Well, this is quite amazing. It is detecting nitrous oxide, and it's really good at it too. So I take a look at the strawberries again. Now I'm being led down a different path, knowing that the nitrous oxide is being produced by excess nitrates. Do I have excess nitrates? Because I've got the data, particularly from strawberries, where I've got light infestations and heavy infestations of the two-spotted spider mite. And lo and behold, the nitrate levels are significantly higher. I would like the nitrate levels to be well below 100, even below 50 if I can get that. The nitrate levels are above 200, and we have low mite pressure. Once we get to 600 parts per million in the plant, we have high mite pressure. So you can see how powerful this is in giving us clues into understanding what the two-spotted spider mite wants, what it likes, and what it dislikes. The IR spectra of proteins. This is a complex science for people who do it, much smarter than I am. They will tell you that the IR spectra of proteins are very complex. And because they're very complex, they're difficult to ascertain when you're dealing with these big proteins, which can be 400, 500 amino acids long. Big, really big, okay? That's not the case, though, with smaller peptides. Smaller peptides, we have, let's say, 2 amino acids combine, and these 2 amino acids combine and form a peptide bond. So you have one peptide bond with a dipeptide. This might have 400 or 500 amino acids. This one has only 2. When you analyze the IR spectra from a dipeptide or even a tripeptide, you can easily detect the peptide bond. On a big protein, you can't see it. So the peptide bond is observable in IR spectra. What does this correspond to? Corresponds to broken down proteins. The mite, as well as other insects too, are interested in broken down proteins. A healthy protein is big and it's indigestible. A broken down protein is more easily consumed by insects that don't have the enzymes to break down a 450 amino acid protein, but they can sure break down the small ones. So when you've got little bits of broken protein in a plant because the plant's unhealthy, two-spotted spider mite is keying in on that. And that peptide bond corresponds to 6,098 nanometers, 6098. So if we go back to the composite graph, there it is. Peptide bond, 6,091. Mites have the ability to detect broken down proteins, characteristic of an unhealthy plant. In addition, did you know that during periods of high heat and drought, plants begin to catabolize, that means break down their proteins, into smaller constituents? Do you see where I'm going with this? They like hot, dry conditions. Plants start to catabolize their own proteins, and this is when the two-spotted spider mite comes in. Everything is lining up. I'm giving you a more complete understanding of exactly what the two-spotted spider mite wants. Here's something interesting too. Bitter pit in apples. It's a big problem in apples, but only recently. Hasn't been around for as long as it probably could have been, but the ag consultants do report a higher incidence of two-spotted spider mite on trees exhibiting bitter pit. Well, what's up with that? Well, here's a paper. Taking a look at the hyperspectral range of bitter pit characteristics in apples. And they found that there are 3 main frequencies which are indicative of bitter pit. So if you were trying to detect bitter pit, these 3 frequencies are the most common ones in order to tell you that bitter pit is present. Allow me now to pull up one of the spectrums for the two-spotted spider mite, albeit in a different range than the one that I've been showing you. 730 nanometers. 1250, close enough, corresponds to 1255, but that's a smaller peak. And the last one, 1410. The 1410 peak, those are the 2 major peaks found in the mid-infrared range of the two-spotted spider mite. So when someone tells me that the bitter pit is detectable by the two-spotted spider mite, I am only going to agree. And I can explain why they are doing that, because they're tuned to it. This is what they're looking for. In this particular case, it's a calcium deficiency. So if you don't have enough calcium, you can go talk to Mr. Ravenberg and he might be able to set you up. But when we have this calcium deficiency, which leads to bitter pit, it gives off certain frequencies that the two-spotted spider mite is keying in on. So here's yours truly. I'm in a cut flower region right now, and this is in California. I've got another ag consultant off to my left. The manager of the flowers is directly in front of me, and this is a quick stopover. This was actually taken during my honeymoon. So before you get the wrong idea, you should know that the photographer was my wife. So it was just a quick stop. I wasn't there for that long. But while there, you can see the cut flower industry is usually in a greenhouse and they have some issues. Did you know that greenhouses prevent about 99% of the ultraviolet radiation from passing through its ceiling and walls? Did you also know that flavonoids, another secondary plant metabolite, and there's over 5,000 of them, are produced only when you have adequate amounts of UV. If you do not have adequate amounts of UV, the flavonoids are not produced. And not surprisingly, the flavonoids are also potent deterrents of the two-spotted spider mite. They don't want to deal with flavonoids either. This is part of the reason why we have outbreaks of spider mite in greenhouses. And just for good measure, know that this is not just about 3 secondary plant metabolites. Glucosinolates and brassicas. The glucosinolates, we have 2 peaks, the 9520 and the 11,600 correspond to glucosinolates too. Another secondary plant metabolite that the two-spotted spider mite is not interested in getting into. If there are high amounts of it, it will be deterred. And that's from a 2012 paper. So you can see where all this is going. It's not just the flavonoids too. Alkaloids in citrus, terpenoids in cucumber and citrus, indole glucosinolates in thal cress, acyl sugars in tomato. Without the assistance of these secondary plant metabolites, plants remain vulnerable to mites. They don't want them. In order to make sure that they're not getting them, they need to be able to detect them. So if they taste them and they're there, which is going to be found in a high Brix plant, they move on. So let's summarize. Let's see what you have learned. Where were you at the beginning of this presentation, and where are you now? Let's give you some conclusions right now. The two-spotted spider mite, it's attracted to drought-stricken plants. Excess water, it's a deterrent. Rainy weather reduces mite survival. Soil with poor water retention, not enough microbes, or inadequate irrigation result in higher two-spotted spider mite populations. You should be able to understand that much better. 2, the two-spotted spider mite is known to attack low Brix plant, published in 2023. Low Brix plants contain incomplete proteins, bad photosynthesis. They're acutely attuned to these proteins through the recognition of the peptide bond. 3, the two-spotted spider mite is known to be attracted to excess nitrogen in plants, which you can detect the ammonium and the nitrates, but it's also going after the nitrous oxide. And so we know that the two-spotted spider mite has gustatory receptors tuned to ammonia, and it's very attracted to ammonia too. What else do we know? We know that the increased ammonia concentration results in higher two-spotted spider mite populations. We learned that too during the presentation. The spectral data also supports multiple families of secondary plant metabolites, which are largely absent in low Brix plants and in greenhouses. And finally, 7, the two-spotted spider mite is acutely tuned to the predominant frequencies associated with bitter pit. So I have been through a lot. I have skipped a lot, but I wanted to give you a sampling of what this was all about. That if I sit down and I decipher the insect olfactory code, or in this case a mite, then I'm now be able to tell what it likes, what it dislikes. Hopefully I'm able to teach some of this stuff to you. You'll be able to understand a little bit better why the two-spotted spider mite does what it does. And the reason why it does what it does is because it needs certain things. It needs to make sure that its cheeseburgers are not found in a big water balloon, among other things, okay? So you are now getting in the mind of a two-spotted spider mite. Helps you to understand what it's going after, and then quite possibly what you might be able to do in order to either reduce the numbers or possibly prevent yourself from ever having a two-spotted spider mite outbreak in any of your crops. So this does conclude it right now. As I said, some of you are going to love this and you just love the data, and that's great if you're scientific nerds. For those of you, this might have been a bit much. I get that too, but I've had the opportunity now to do a deep dive into a particular insect, and maybe, just maybe, some of you won't ask me too many more questions about insects, thinking that I'm going to start to give you this. But we'll see. All right, thank you very much. Go ahead, I can repeat the question. Yeah.
Just thank you. Your patience level is way above mine.
Way above yours, yeah.
Thank
Way
you
above,
for that.
yeah.
Question is, the life cycle of the spider mite play a part in any of this that, in your research?
Yeah.
Or what is the life cycle of the spider?
Well, the life cycle of the spider mite, it plays a part because they're everywhere in small numbers, just kind of waiting, watching and waiting for things to happen. And when the conditions are right, they explode. So the life cycle is dependent upon them with getting that high nitrogen content source that they need, among other things. They need to make sure that there's not enough water because they're trying to eat as many cells as possible. All of this has to do with the life cycle, because if these conditions are not there, the life cycle's broken and it can't reproduce. And they will just hang out on your plants indefinitely, waiting for the conditions to just be right. And in some cases, in some years, you know this as well as I do, sometimes you don't get an outbreak with a two-spotted spider mite because they have to have all the conditions correct. So their life cycle is absolutely modified. Is this for me? Did I just lose? The other one went off. I'm not sure if you were hearing me from the back. I apologize why the other one went off. But so yes, this does lead into the I never liked him. So that's a good question. Any other questions? Yes.
Would these mites start at the bottom of the plant or at the top?
they're going to be neither at the top or the bottom. They're usually going to be on the underside of leaves. If the leaves are at the top, they'll be at the top. If they're at the bottom, they'll be at the bottom. They're not found around the stem. They don't feed upon the stem, so they're usually found on a leaf. Some of them will be found on the top. Most of them will be found on the underside. The reason why, I didn't want to mention this because it's just another detail, but they prefer the parenchyma cells rather than the palisade cells. So the parenchyma cells are on the bottom of the leaf, which have the chloroplasts, which they're interested in. The palisade cells, which are also photosynthesizing, are at the top, and they're not as interested in those. So you usually find them on the bottom, on the underside of a leaf, and they can be anywhere where the leaf conditions are adequate. In other words, low secondary plant metabolites, high nitrogen, and then they will start to feed, and they'll feed as fast as they can. They'll keep feeding until they get full. And as I said, they can suck the contents of a cell in a matter of seconds. So that means they're going through several cells over the course of a minute. And this is why you see stippling. When you see that characteristic stippling in your leaves, those are individual bites of the two-spotted spider mites, which suck out the contents of the leaf, cause a brief discoloration, and that gives it the stippling effect. So, wow, good question. Any other questions? Cindy, I know why there are no other questions, because lunch was just brought in, and people are thinking, please, just get off stage right now. I want to eat. If that's the end for me, then I will just hand the mic over, and we can start to eat. It's all yours. Go get 'em.
Thank you, Dr. Dykstra. Very much appreciate this.